Thank you very much for the warm welcome. I'm starting to share now. I hope it shows up. Maybe it takes a minute to refresh, but I think we're there. Just let me know if anything, I don't know, if there are glitches or if you don't understand anything.
We are seeing your speaker view, not your
Yeah,
slides.
Oh, that's
you are
always
showing the
okay.
presenter
Give
view
me,
different than yesterday.
yeah, yeah. Give me a second. Yeah, the numbering of the screens is the other way around than I would have expected. So now you probably see
Got,
the actual
yeah,
screen.
That looks good.
Okay, perfect. So, 3 words to myself. I'm Anna. I'm a neuroscientist by training, and since 2012 now, I'm working as a product manager at Zeiss for the multi-SEM, the world's fastest scanning electron microscope, namely a multi-beam scanning electron microscope, which I'm going to talk about a little bit now. So, in a nutshell, as I said, it's a multi-beam scanning electron microscope. That means it operates with 91 beams in parallel, which can scan up to 80 megahertz, which would be 12.5 nanoseconds in dwell time. And therefore, in the end, the top net speed or the top net data throughput would be about 3 terapixel per hour or 3 terabyte per hour. So if you can probably do the math, what that in the end means for the IT infrastructure of your institution, but can come to that later. The multi-SIM comes with a spatial resolution of about 3.5 nanometer, and overall it's designed for ultra high throughput operation. And that means basically 24/7 operation. It doesn't make too much sense if operators need to interact with the system all the time to shoot a couple of images ultra fast and then do something again. So everything is set up in a way that it runs mostly automated after you set up a longer term experiment. As a scanning electron microscope, obviously it's capable of imaging not only life science samples or ultra thin sections, so it's also capable of imaging chip samples from semiconductor research or all types of material samples. However, our main customer base and our main market segment, so to say, right now is connectomics within the neuroscience field. And that is also what I will talk about later mainly. So just to set the space, to set the stage, a couple of words about volume EM. You probably know already a lot about it. I just wanted to point out that it is an emerging field or emerging trend since a couple of years. So in 2023, Nature judged it as one of the technologies to watch. And when one has a look at the number of publications coming out every year with either array tomography or volume electron microscopy or serial block phase scanning electron microscopy as search words, we'll see that the number of publications is growing each year. So 2026 just started, so obviously there's not so much yet. Now, this year, or basically last year, electron microscopy-based connectomics became method of the year. Again, out of the Nature journal. And now, if we, as a subfield of volume EM, if you have a look at the subfield of volume EM and the number of publications coming out here, it's basically the same pattern. The number of publications is growing year by year. And when we think about connectomics as a way to elucidate the connectome, so the overall network of a neural system of an organism, by now, in the past, it was only possible to do that on a very, very small scale. So basically, the level of detail for small animals, flies or worms, or maybe only tiny portions of larger brains. And we think that now the time is ripe for high throughput technologies, which are capable of capturing more of these brains than just these tiny portions than about a cubic millimeter, which in electron microscopic terms is still quite a lot, I must say. So there are various methods for volume EM, namely array tomography with a single beam system, serial block phase imaging with a single beam scanning electron microscope, and also focused ion beam microscopy, which all have their advantages and disadvantages. So all have their niche where they're probably the best solution. And amongst these, multi-beam array tomography is actually just the upscaling of array tomography, which makes it just faster, more automated, and allows for larger areas and volumes to be acquired. Now, why is speed such an issue in this field of connectomics? Now, the level of detail one would need to resolve in brain sections, in brain samples, is actually fairly small. So vesicles are about 60 nanometers. The membranes are like, I don't know, 10, 20 nanometers maybe. And the axons can be also very thin. On the other hand, they can span large distances, like maybe the most extreme from left to right hemisphere, but also within a hemisphere or even within a certain cortical area. The neuron can extend over a millimeter easily. So to capture a full network, like a full mini connectome within an organism's brain, already requires quite large volumes to be imaged. And if you do the math now for a cubic millimeter, well, the standard way of working, or the standard application, I would say, for array tomography then would be to cut this tissue into thin sections. I would say 50 nanometers is like the average. People tend to use as thin sections as possible to get a better Z resolution. So 30 nanometers is maybe the lower limit. But let's calculate with 50 nanometers. We will end up with 20,000 sections, namely 20,000 square millimeters to be imaged. And with a single beam electron microscope, only 1 square millimeter probably takes about 2.5 hours, depending on, of course, on the parameters and everything. Just again, a rough estimate. So imaging that amount of tissue would sum up to almost 6 years of just pure imaging time. And in principle, with something like the multi-SEM, this can be reduced to just 1 month now. I already mentioned the extremely fast scan speed the multi-SEM can do. That, of course, is only usable if the sample contrast allows for that. So can come to that later again. So that's, I would say, the extreme minimum. But still, 1 month to 6 years is like a huge, huge advantage. Now, this comes with a handful of requirements for the sample. I said if the sample contrast is good enough for such fast scanning. I can share these like full slides with the references to the papers. This is something I can easily share later, so don't worry too much. You can't read everything so quickly. In general, the standard sample preparation protocols for volume electron microscopy, namely for serial block phase imaging, are usually very well suited for the multi-SEM as well. So the samples should have a lot of heavy metal contrast. They should be as conductive as possible. And this is also true for block phase imaging. There in the meantime, quite a number of publications out there that describe the best suited sample preparation protocols for these type of workflows. Also, when it comes to cutting of the sections, the collection of the sections, besides the, in the meantime, almost standard tape collecting method, another method entered the stage, the so-called MAG-C or Gauss-EM, where sections are collected not on a tape, but directly on a wafer, directed with like magnetic help. And all these techniques have one thing in common. They are very quick. They can be automated, and they can generate a lot of sections without the need to manually cut everything and place it on the wafer. So for us, there are like 3 sample preparation, mainly automated sample preparation workflows that our customers are using. The first one would be this magnetically directed section collection on top of the wafer directly. The one on the lower, that is non-commercial. This is something that a handful of our customers are working on to get a fully integrated 3D ablation and imaging system. Something like a FIP-SEM, where the ablation technology is placed besides the microscope. The picture on the lower right maybe gives an impression of how complex such systems then are in the end. And in the end, most of our customers are actually using the tape collecting mechanism, the atom tome, and that's also the type of workflow I will show now. This is maybe the most well-known one. The others are a little bit more recent and not that established yet. So to give you an idea how such a sample could look like, so this is one of our sample holders. It's a little bit larger than 10 by 10 centimeters. So the sample area can be 10 by 10 centimeters. And what we see here is a square piece of silicon wafer with tape strips on top and serial sections placed one after the other on top of that. And the first step in multi-SEM imaging is always placing the sample under the light microscope first and generating an overview image. So that would be a screenshot from the light microscope while acquiring an overview image of this full sample. And that in the end means that a typical multi-SEM lab actually has not only the multi-SEM, but also a light microscope, which is, we have a laser here somewhere. Yep. So this would be the standard light microscope we use together with a multi-SEM. And this gives you maybe an idea of how such a lab would look like or could look like. So it's a little bit larger than a standard microscope. I agree. And it also comes with a little bit more of like side equipment, like these 2 racks here in the background. But on the other hand, it's still fitting in like a standard lab space, I would say. Now, when the sample has been in the light microscope and the overview image has been generated, the next step is to bring it into the multi-SEM by means of a standard airlock, which is also commonly used in single beam systems as well. And this now is a screenshot from the multi-SEM user interface. And you may realize it looks very similar to the one that we just had from the light microscope. This is the reason for this is it's basically the same software platform. So the user interface in both cases is Zen. So it's pretty easy to accommodate to the usage of a multi-SEM if one is already used to light microscopes from Zeiss. Now, after the sample has been introduced to the multi-SEM chamber, setting up the experiment is guided by, or can be guided by, something like an experiment setup wizard, which guides you step by step through these different steps. And the first one would be to define ribbons in this type of sample. I'm jumping through the video a little bit, that it's not going too long. And the second part would be to define an individual section as a template. And when that section is defined, the software is searching for these shapes on the rest of the image, and then all the sections are detected. And the next step would be then to define a region of interest. If you don't want to image the full section, but only a smaller area, then this is how you would do that and apply it to the rest of the sections. And then the next step already is transferring all these regions now into the stage coordinate system of the machine. So right now we're only working on the on top of a light microscopic image that has been imported to the software. But now we're transferring these coordinates to the stage environment of the multi-SEM. And the overview image can be added to that view as well. So it's really easy to check if the regions of interest are positioned in the correct areas. To be able to have all of the data in the end nicely focused and without astigmatism and everything, we then set up a focus map, like an autofocus workflow, where a number of focus support points are distributed for each of these areas. And then you can just pick out one of these regions of interest and distribute this focus support points wherever you want. Usually we recommend to put them outside the region you would want to image to avoid contamination. And then again, these settings are copied to all the other regions. So no need to go through all these, 200-300 sections that are on such a wafer. And then when the experiment setup is done, the experiment can start, and the system now jumps from one focus support point to the next, checking for focus and astigmatism. These values are saved, and in the end, a plane for these values is calculated through these focus support points, and then the imaging starts. And in the beginning now, it's zooming in a second. So it's jumping now from hexagonal shape position to the next. This is actually real time. So this is in principle how quick one can imagine such an area to be imaged. Now it's speeding up a little bit, so let's jump through. So this is about a square millimeter. Now, what is actually happening? We just saw now that hexagons turned green and that image data popped up in the other window. But what is actually happening? What is the multi-SEM actually doing? So we're starting with a single beam, as with any other electron microscope, scanning electron microscope as well. And this electron beam is split with a multi-aperture into, in our case, 91 beamlets. And these beamlets are then scanned over the sample surface. The secondary electrons that are generated are collected and projected into a second column. So we have two columns on our multi-SEM. And these signal electrons are projected onto a scintillator. And this is the actual signal. So in the end, it's a photon signal that is picked up by optical fibers and then detected by avalanche photodiodes to generate the images. If you would compare a multi-beam electron microscope to a single beam electron microscope, it actually becomes pretty clear what the advantage is. The field of view is just way larger for a single scan pass. That's basically the whole trick. And as we already saw in the video before, by moving the stage, we can acquire even larger areas, basically by mosaicing this full sample areas. If you're interested in viewing this full workflow in a little bit longer and with more explanation and without jumping in between a little bit more detail, we have a virtual demo that was part of a science booklet a couple of years ago. Again, I can share the slides with the QR codes like afterwards. So this is this can be checked later. Now, what comes out of it? I mean, that's maybe the main question. One of the major publications that came out of one of our customers' labs was a science paper from Jeff Lichtman, where a petavoxel fragment of human cerebral cortex got reconstructed. Petavoxel means it was a 1.4 petabyte data set, and in volume it was about a cubic millimeter. So it was not like a cube, it was more like a slab, but still the volume in the end was a cubic millimeter. And this got fully reconstructed. It's also accessible online. One can play around with the Neuroglancer tool there. So I also would highly recommend checking out the paper. It's really nice. And just gives you an idea of what could come out of such a data set in the end. So here it's only the excitatory pyramidal neurons that are rendered. Or if we could go also in a little bit more detail. Here it's an individual pyramidal neurons with synapses and axons labeled. So the wealth of data that is available here in such a data set is pretty incredible. And still, it's only a stepping stone, I would say. If one looks for landmark connectomics projects, the first one probably was the C. elegans connectome, which was acquired back in the 70s and 80s, like a 16-year project, as one see. And C. elegans only has 302 neurons. So this is really a tiny, tiny, tiny little connectome. And the next step then was already Drosophila, which is way larger in comparison. The brain of a Drosophila is order of magnitude larger than the roundworm. Still, in the end, the amount of data was just tens of terabytes. So now with this petabyte data set, there's also one from the mouse brain, from Allen Brain Institute. The cubic millimeter is now something like in between, and again, an order of magnitude larger than the connectomes before. However, that's not the end of the story. Right now, a project is ongoing that has been funded since 2023, where the mouse hippocampus shall be reconstructed, which in the end would amount to about 25 petabytes of data, so 25 times larger than the data set that are available right now. And ultimately, researchers are aiming for the full mouse brain, and then, of course, in the very, very end, maybe the human brain. But even the mouse brain, the full mouse brain, again, would be an extraordinary effort, because in the end, it would result in an exabyte of image data. And well, it's pretty difficult to wrap the mind around that. 1,000 petabytes of data, 1 million of terabytes. Still, there are two institutions that are wrote up proposals for such a project. One came from the NIH. As I said, this project has in principle already started. And another one has been formulated by the Wellcome Trust in the UK. So by now there are at least two larger institutions that at least think about something like that. Besides this huge data sets that can be generated with a fast electron microscope, there are, of course, other projects that could also be done with something like that. Like other of our customers are rather looking for smaller data sets, but more of them to for example, compare the brain structure of mouse, macaque, and humans, or to elucidate a cortical column. Again, a lot of method papers are out now, how to deal with that amount of data, how to process it, how to handle it, or also just how to prepare samples that are usable for such projects, because it's also not easy to collect section series beyond like 1,000 or 2,000 or 20,000 sections. If you need to collect this number of sections, but without losing anything in between, this becomes another challenge. So to wrap things up, in principle, all that I've showed so far was done with the Zeiss multisem, with the old Zeiss multisem, the 5 series. So right now, we just launched last year the multisem 706, which is basically just a next generation. It's I would say it's kind of an evolution of the older system, with a faster scanner, so we can reduce the pixel dwell time and therefore scan even faster and maybe increase the throughput even further. The scan electronics is much better, so stitching is much better. The images fit together much better, which you can imagine if you're not only mosaicing by moving the stage, but your field of view is already a mosaic of 91 individual images. This is an actual challenge. A little bit more technical, landing energy range has been a little bit increased. The scintillating material became better, so the mosaics are more homogeneous, basically look nicer. The system is more stable than before because we exchanged air cooling by water cooling. So yeah, as I said, it's not like a revolutionary change, but it's more like an evolution. And with this, I would end now and be open for questions if there are any. And
Hi,
thanks for
Anna.
your attention.
Thank you. It's quite impressive. We have 2 questions from Stephen. Stephen, you want to speak out to ask a question directly? He's listed. It's in the chat. One is about the sample preparation, and I don't know if you can see
Okay.
it. And
I'll just check the chat.
Stephen, are you here? Yeah, please go ahead.
It says, hi,
Yeah.
I wonder about if standard osmium oxide post-fixation is enough for good contact on the sections, or OTO is necessary. And then contrast. He meant to say contrast.
Yeah, all good. I would recommend the OTO or even the reduced osmium, the ROTO or the ROTOTO protocols. It's always a matter of trial and maybe error. It's really hard to tell in advance. But what we've learned is the more contrast, the better. So the best results we get from the ROTO protocols, which then some labs even worked on these and added some other nasty chemicals to even improve the contrast. So yeah, I would say yeah, rather more contrast than less. And
so there's also another question.
There's another question. Apart from brain, which organs do researchers reconstruct with volume EM?
Yeah.
So by now, it's actually mostly brain. I believe that has several reasons. I think first is that brain research were already prepared for something like that before such a system existed. So the idea was to use multiple electron microscopes in parallel. In some workflows are actually doing that. So the whole data processing pipeline was already kind of set up in a scalable way. So in that sense, here the match was easier. And the second thing that is more technical. Brain tissue is, I think, actually maybe the easiest to work with because it's so dense. You have so much fat in there. It can be stained so nicely. It can be cut so nicely. So with other tissues, I think it's more difficult, and also maybe the volumes don't necessarily need to be that large because the tissue is more homogeneous. On the other hand, we're really interested in learning about other potential applications and other tissues that could be imaged. So why not?
Okay,
So,
we have one more question. Tom, you want to read it? Or so it's about the new software supports for real-time stitching across different field of view and the brightness normalization.
Yeah, significant illumination differences. What might be causing this, and what methods can be used to address it? Oh, 505 user. Hi, non-neural tissue samples. That is actually hard to answer now on that, well, cut like that. Well, I would say contact our application support or the service support, because all in the system itself, it of course always can be a technical reason. On the other hand, we have a functionality that equalizes the detectors, and that of course should be used from time to time, and everything needs to be set up nicely. When the question is now for the new software, it's not necessarily the new software that enables the brightness normalization. Is actually the scintillator that is much more homogeneous. So the issue doesn't really come up. And then, well, I would say most of our customers of the older systems are actually post-processing the data. And to equalize the histogram across the image tiles is a pretty basic image processing bit. So that we probably could also assist with.